calcium-responsive liposomal nanoparticle reporters. So this slide explains about the problem with current calcium imaging technology. So on the left side, you can see calcium optical calcium sensors, which has very high sensitivity, but it has limited depth. On the right side, you can see that we have MRI, which provide whole brain coverage, and you can sense molecules deep in the brain. But the problem is they have poor molecular sensitivity. So we overcome this problem using a liposomal nanoparticle reporter. We call them LisNR. So the mechanism of LisNR, how it works is it's here on the left side. So they are liposomes which enclosed MRI active contrast agent. So we play with the membrane permeability. So there are different ways to play with the membrane permeability. If the membrane is less permeable, so water cannot go in, or the exchange rate is lower, so you have darker MRI image. But if the membrane is more permeable, so water can go in and out, so you get brighter image. So one way to do is you can have a light responsive lipid. So and you can also make the pore in the membrane. So the third one is we make calcium responsive lipids, and they change its conformation when they bind to calcium. So this is the one I'm going to talk about today. So this slide explains the whole we validate our sensor both in vitro and in vivo. So you can see on the left side, sensor responds sensor responds to calcium, and it has no sensitivity for magnesium, and it is also sensitive in more physiological medium like ACSF, and when it is supplemented with some protein. And this is also reversible, and also at 37 degrees Celsius reversible when you remove calcium or supplement calcium, so over multiple cycles. So on the right side, so what we do is we artificially manipulate the calcium concentration on the brain. So what we do is we inject a small molecule called EGTA. It is a calcium chelator. So what we do is it actually chelates the calcium and decrease the local calcium concentration. So you get a signal which is less brighter. So the trend is all across the animal. You can see in the green color is called Cas LisNR. And the liposomes on the other side is a control which has no sensitivity for this. So what we actually, the most interesting part here was in EGTA side. EGTA is a small molecule. So when you monitor this side over a long period of time, so what happens that calcium response, the sensor actually activates again over the time. And this was also observed across the animal. So next, what we are trying to do is we are delivering the liposomes all across the brain so that we could map calcium activity throughout at whole brain scale. So we use different methods to deliver the calcium responsive LisNR. As you can see here at the bottom, we were able to actually deliver these sensors. And now next we are trying to use stimulus, like more physiological relevant stimulus to actually map the calcium dynamics in the brain, with the hope that we could actually map neural activity more directly than more conventional fMRI based. Thank you.